nothing on sonar yet, so it must be pretty relatively flat. Yeah.
Inside. Cool.
Yep, we have
Still
definitely
coming
found
down
the
at
bottom.
15.
We got some rocks there too.
Yay! It's a rock.
Copy that.
Very excited. All right, so for those of you who are just joining us today, we are at Pigafetta, Gio. We're all trying to pronounce it correctly. Pigafetta. Gio, and we are at a depth of just about 2,000 meters. And what we are hoping to do today is move from north to south along a ridge, and we are going to be looking for manganese-crusted rocks and all sorts of exciting biology.
Of which we can see some already.
You're right.
Yeah, Delta
And a
20
rock.
still coming down.
Oh, and there's a fish. Oh, and an urchin.
Yeah, I was going to say,
and
the
a
urchin's
coral.
very
and
cool.
corals, yes.
Very exciting.
I'm stopping for a second, Pod. It's got some heavy
So this is a
leaves.
synaphobranchid eel. We've seen quite a couple of these on this expedition so far. Also known as a cutthroat eel.
Holding.
Thank you, Bruce, for that ID.
Boy, those are cool.
It's got quite a bit of scarring.
Oh, what is it doing?
Bridge nav.
It's got quite a bit of scarring on its on the side of its body.
Yep.
Go ahead, Nev.
Hey, just letting you know we're on bottom. Our bailout plan stays the same at 291 degrees.
You are on bottom. Bailout is the same at 291. Good copy. Thank you, Dad, for letting me know.
Thank you.
Have a good dive.
This is Bruce at the IRC, fishery biologist and ichthyologist who study
Nice.
fishes. These
Zoom it out.
Delta about 15.
very dominant, but they're also
Wide.
scavengers.
Yeah,
I
we
often
are
see
seeing
them with this
a
type of
huge
scarring
side,
on the side, and I think that
surprised.
maybe because of their active habits of feeding, where they get into things where they
At least we were.
might scar up their skin as they dig in for food, or maybe it's even a reaction to some of the crustaceans, damage from some of the crustaceans and things that they might feed on, although that seems less likely. Now you've got a nice urchin and some corals, so I'll be quiet and let the coral biologist jump in.
Thank you, Bruce.
Thank you, Bruce, and good morning. So we've also got, a ophiuroid has just leapt off of that coral and collapsed onto the rock. So we can also see this big, beautiful coral right here, as well as it's got a couple
Yes.
commensal
lasers are
ophiuroids
coming
and
on.
anemones, and
Hold
then we've got this fantastic urchin or echinoid.
Lasers on.
So this is likely an
finish with the urchin, if it would be possible to look at the bamboo coral and particularly where it's branching and perhaps a couple polyps.
okay, great, sure. So this urchin is likely an echinothurid, perhaps from the genus
Zoom it in.
Trimicosoma,
Holding.
which is also seen off of Hawaii and the northwestern Hawaiian Islands as well. So, Pilot, when you have a chance, if we could have a look at the coral as well, that would be brilliant. Thank you. Pilot? Okay, just making sure you heard that. When you have a chance, we'd like to look at the coral as well, please.
Okay, zoom it out.
So we're getting quite a couple of reports from shore that the video connection is quite bad, and we apologise for that, but I think right now it's probably a lot due to weather, because we're not having very good weather.
It's not just
out
weather.
here. I know, but... So this is a big bamboo coral, also known as an icidid.
Zoom it in.
And we're going to get a closer look at the polyps and the branching to see if we can figure out what genus this is from.
Looks good on my monitor. I'm going to zoom in a bit more. Full zoom.
Beautiful. Thank you,
so
Pilot.
unfortunately, Les and Scott aren't on at the moment. I'm a little uncomfortable with these large, bushy bamboo corals going to one of their clades or some of the genera that they've described. But it looks like it's even hard to tell whether or not it's branching at the nodes or not. So I think I'm just going to give it a ice it and leave it at that.
Okay, sure. Chris, thank you. Okay, pilots, that's great. Whenever you're ready to go, we are also ready to go.
No, we want to pick up a rock.
Okay, well, whenever you're ready to pick up a rock, we're ready to pick up a rock.
Full wide.
We'd like to try to pick up a rock here before we start moving. Always. I think you've hit it all. So, Patty, if you want to scream out if you see a rock that looks particularly good while we're searching.
Okay, this is Patty Fryer, UHECC, marine geologist, and I am looking for a good rock. Most of these platy things, I'm suspicious they might be indurated sediment. So if we want something igneous, we want a blocky rock. So,
right.
Angular, I mean.
Yes. It does look sort of blocky, but we've been fooled before. Try to squeeze it. Yeah. I mean, not so much that, you would totally destroy it, but if it's sediment or indurated mud or something like that, it's just going to crumble.
Go for it. You got an opening.
Yeah, it looks like a good target.
Yep. And while we're looking at this rock, I'll just note that this looks like there's a lot of manganese crust around here. You can see that botryoidal shape where it's just sort of that round and pebbly looking. So my guess is we're fairly we're pretty much looking at all sorts of manganese crust around here.
Sure. 180, 10 meters. Okay. Hey, bridge nav.
It's about as low as I want to get, though.
Go for bridge.
Can we get a ship move of 10 meters at 180 at 0.2 knots, please?
Yep, you were
10 meters
spot
at
on.
180 at 0.2 knots. Good copy.
Thank you.
Zooming in.
Yes.
Holding.
Okay, great. Yep.
Zooming out.
RV nav bridge.
Fantastic.
Go ahead, bridge.
I'd like to come 15 degrees to starboard. My heading.
Copy that.
Perfect. So then let's try to get some pictures of this, and we'll take it with us.
All right, pilot, just be aware if you're going to put it in your port, you need to open the lid.
That's a good rock, guys.
Box, you need to
I
open the
agree.
lid. Yeah, I just wanted a heads up before we move it over.
Zooming in. Holding.
Are any of those biology?
I mean, they look like hydroids flock, and I'm not sure what that big one is.
Zooming in. Holding.
Yeah, a little sponge.
Cool. Great. Can we zoom out a little bit and kind of rotate the rock a little bit? Thank you. I know.
Zooming in. Holding.
Yep. Yep. Diva, you're going to have a great time with all the commensals on this today.
I mean, I think, yeah. I will. That's
You will.
for sure.
We have arrived at our new position.
No, it's
Copy.
fine. Just
No.
put it in the rock box.
I agree.
Okay, zooming out.
Yep.
Full wide.
I would think it would be okay
Yeah,
in the
I think it's
starboard
fine.
box.
Cool.
Yeah. It doesn't look that small, so...
Pilot, do you want the wing inboard, starboard? All right. Starboard wing coming inboard.
I'm going to change the location.
There it goes. Copy. Do you want any light adjustment? Do you want any light adjustment? Job, pilot.
Great,
Starboard
thank
wing
you, pilot.
coming outboard. Close the drawer.
All right, so we just collected our first rock sample soon after getting down to the bottom. We've had a chance to look at a little bit of the biology around here. And I think our next move will be to start slowly moving up the ridge and trying to see what we can see.
Moving when y'all are set up.
Chris, what are you particularly excited about in terms of seeing today? What are your hopes for biology?
Yeah,
Well, my hopes is that we see a lot of corals and sponges. This is a pretty good start. We're seeing a bunch of colonies. I wouldn't call this a particularly dense community yet,
Right.
but there are definitely some corals in the distance that we can see, and saw some coming down. And if, we'll try to document each type if possible, so we get an idea of what the components of the community are. And then just keep moving up, and I think as we get closer and closer to the little pinnacle on the top, I think they're going to get denser and denser. So we may wind up with a really spectacular community at the end of the dive.
Great. And Chris, could you introduce yourself for people who are following along?
Yeah.
Yes, I'm sorry, I keep forgetting to do that. I'm Chris Kelly. I'm the program biologist for the Hawaii Undersea Research Laboratory here at the University of Hawaii, and I'm serving as a science advisor here for the Okeanos 3-year effort in the Pacific.
Thank you very much. And so I think what we're doing right now is setting the ship up to start moving. so that we will then be following it as we move along
Ridge
the
nav.
ridge.
And Chris, I think looking around the
Go ahead,
seafloor
Dad.
here, I've noticed
We
quite
get
a couple of
ship move
the
of 80
really
meters
large
at
sponge
170
skeletons
at
dead.
0.1 knot,
Where you
please.
can see the stem with the sort of striations coming
do
off
it
it. I remember the
upper
first time we
surface.
saw those
80
in
meters, bearing
off Hawaii,
170
we
at
were
0.1 knot. Good copy.
confused as to what they were, but now we know. But so I guess that kind of bodes well for
Yeah.
further up the hill, right? Oh, and I think
On
I can
this
see one
flatter
of the
terrain,
sponges
it's
I'm
nice to have
referring to.
Oh, there's
further
already a
up
bunch
the
of stuff
all the way
back there,
up.
yeah.
All right, pilot, I'm going to put the uppers out. Real quick.
Yeah, in fact, I'd probably go for that tall target in the back if that's in the direction that we're headed. And then there's another bushy bamboo next to it. There's a bunch of things that would be good to document before we leave this area.
So, pilot, before we start moving, if we haven't started moving, we'd like to document a bunch of these guys.
Yeah, I figure.
Are these alive?
Yeah, little frogfish.
Yeah? Okay.
So,
Yeah,
so
sure.
this
Let's
particular,
zoom on the
I'll
fish.
start with the sponge first. This particular sponge, when we first came across it in the Northwest Hawaiian Islands, we called it the leaf sponge because it looks like a big flat leaf. And then once we were able to collect the specimen and identify it, look at its microscopic components, then we were able to identify it to genus. This is something that's called Tritopleura, and it's in a sort of a weird family called Incinitaridae, and it's a hard sponge. It's very brittle, and so it's kind of interesting. And we've also got a really interesting fish right here on the bottom as well.
Yep, can we
So
zoom in on
before
that?
the sponge, we can get the
Yeah,
fish.
perfect.
Oh, and a holothurian
Yeah.
in the background.
Okay,
So the
thank you,
fish
Rick.
is from the family Chornosidae.
How cool.
And it's likely Chornocops coloratus.
We saw this the other day at the pillow events, right?
Was it this exact same species?
It looked very similar. Oh, I'm getting the nod from another biologist. Yeah. Yeah, it was the one that was hiding inside the pillow mound.
Yeah.
You don't believe me, do you?
Zooming in.
No, I remember. It's just...
Holding.
So we can see it's got its modified fins that it tends to sit and walk along on the seafloor on.
That's awesome. I do like these
Can you
fish with
face the
legs,
camera?
which I know is not the right term for them.
And these are also known as coffin
No,
fish
I'm
or
just
sea
teasing, that's fine.
toads, and they're a relative of
All
the
right, that's fine.
anglerfish. Thank you,
Zoom in.
Bruce, for that.
Should saw a speaker in detail.
Can we do a quick zoom on the cuke over on the right, please? Before we get to the sponges and coral. So this holothurian or sea cucumber looks like perhaps Hansenothuria. Need to get a better look at it.
Zooming in. Holding here.
Yeah, I'm going to go with Hansenothuria on this one. Possibly Hansenothuria benti. Great, thank you guys.
Zooming out.
Okay, so back to that sponge, the leaf sponge. So the sponge is likely, as Chris said, from the genus Tritopleura.
So both of these sponges are the same species, just you have one that's older and one that's younger.
Yeah, that would be great. Thank you.
Zooming in. Holding.
So is that the sponge that Chris was just talking about?
Yes, exactly.
How beautiful. Have we seen one of those yet?
No, we have not during this expedition.
Yeah, exactly. This is the first time we've documented this genus here in CNMI, so this is terrific. And I'm also seeing a lot of similarities initially with this type of community in Hawaii, these really big bamboos. We were seeing these at this depth in Hawaii as well.
Chris, and also, did you notice behind the sponge, it looks like there's one of the cladorysids that we collected, the potential new species?
They said they did
where
want
is
to,
it?
like, go
behind
through
the leaf sponge.
There's a coral that's kind of behind it, but I don't see the cladorysid.
They're talking about that one back there. Yeah. Oh, down. Sorry.
The little white thing?
That.
Oh, not the
it's
little white
in the
thing.
frame.
Oh, the little fence thing.
It's way in the background, like to
I'm
your
going to
left,
stop helping,
down left.
obviously.
No, below that, it looks like a coral.
So this sort of leggy thing that's at the bottom of the screen, this is something that's called Candidella, the primnoid, and it's Candidella gigantea. I'll potentially go right to species on that one.
It's like above the lasers now.
The one at the bottom left?
Start moving yet?
Well, these are the ones with a slight curl that are kind of right in the center right now. You know, they're single unbranched, thin little stalks with just a little curl at the top.
Okay. Yeah, there's not much delay.
No, I take it back. Maybe that's a coral.
It's the one to the right.
Yeah.
So the tricky
That
thing
right
about
there,
these is Candidella gigantea has 3 polyps per whorl. And we were seeing some Candidella in the really deep dives that had only 2.
Want me to zoom out?
It's hard to tell from this distance whether that has 3 or 2. I think it has 3. So I think we could be safe in perhaps going to gigantea as the species.
Chris, which one are you referring to?
This is the coral that's right in view right now. This is Candidella gigantea.
Okay. Thanks. Okay, that's great, pilots.
So
Thank
it's kind
you.
of interesting the way the naming came out. This is Steve Cairns who named this from the Smithsonian, if I'm not mistaken. And I believe it was Amy Bacco-Taylor who collected the type species. And the gigantea, the species name, is actually in reference to the size of the polyps, not the colony. It certainly is not a gigantic colony.
Jeff, when we have a minute, be alright if I redid the black balance? Just, it looks like it's shifting green. Okay, I'll do it quickly here. Let's try it here.
That's okay. Do what you need to do.
One more time here. Yeah. Alright, we'll see how that looks when we get closer to the bottom. Thanks.
No. You kept moving forward, so I kept giving you more time. .1 knot. Yeah, I don't, unless we stop and really start doing stuff, I'll just keep it moving. You might even be, nah, I don't know. I thought you might have been pulling zeros, but I don't think you're quite that far out anymore.
Yeah, I think it's just the heave that
Yeah.
Cirrus is feeling.
It's a good one today.
Yeah.
Oh, that's neat. Bottom right there. Haven't seen that.
And this is just essentially a coral garden we've plunged down onto. We can see
That's really
primnoids, isidids.
Yeah, you're
A lot of these
pretty-
corals have commensal
I am moving.
ophioids living
I
on
probably have
them. There's what looks like a Brazingid sea star just going off camera. We've seen some holothurians, some
roughly.
fish.
Yeah,
And I think this is just sort of a tease for what we're going to see later on in the dive, to be honest.
Pretty regularly. It's turning just from the heat,
Yeah,
I guess.
this is sort of
Yeah,
a
you can
start
ascend it right now.
that I'd hoped for, but you never expect it. When we're seeing this many corals
pretty
right
good
off the
ascent
bat,
too,
it's a
so-
really, really good sign. So there was a very old Cretaceous seamount in the Northwest Hawaiian Islands, which had a somewhat similar stand like this, some very large bamboo corals on it, and it was almost a monospecific bed there, in that there was a lot of the same types of corals that just went on and on and on, a little bit like some of the shallower beds that we saw the other days, and still trying to figure out what happens to create these communities. So a lot of people are really interested in that question.
Is that another urchin down there? Cool.
So I see that Scott's on the chat now, and I wonder if we could get a close-up of one of these really big ones here, since he could absolutely help out in identifying these things.
Yeah, but I think he said, Chris, that he's not going to be able to focus on the dive right now, which he was quite upset about, because obviously it's like an isidid. Yeah, an isidid field.
Yeah, I think we're going to see plenty of these today, so we can wait until he is able to take a look.
Yeah, probably. You are getting a little, might be all your tether, only a little left, so you might want to do some side to side.
Sure, I can do that. You want that, or do you want to go exploring more? I'll pick it up 50%. 2? Bridge, Nav.
That's
Can we go
all
ahead
right.
and pick up our speed to 0.2 knots, please? Thank you.
So, Chris, do you know how old some of these corals might be? I'm guessing because it's such an old seamount, many of these animals may have been here for quite some time.
Zooming in.
Yeah, so if I'm not mistaken, some of the isidids that have been dated so far are a couple hundred years old or so. They're not like the really, really old ones, I don't think, such as the Leopathes, the black coral, or the gold corals, but they're in hundreds of years age.
Great, thank you, Chris. So we can see here this isidid has actually got a chirostylid squat lobster living on it. I think it's chirostylid. And also we can see some anemones and also some hydroids. It's always incredible because we see these, what are essentially fields of coral, and then where there does appear to be a lot of life, but then when you actually focus on one coral colony, then you see that that in itself is its own community, hosting a range of different types of animals just within that coral. There are also some barnacles living on this coral as well. as well as I think a gastropod or snail. Pilot, what's the current like here? Okay. Right. I was just asking because there's a lot of coral around here, so was wondering.
Go ahead. Yeah. Okay. Yeah, it looks like we're going to shift over to probably 190 or so. Copy that. I was going to let this finish because it wasn't that far, but if you want to do it now, I mean, we absolutely can. We've got 20 meters left.
So, geologist, does anybody want to hazard a guess on what we might be looking at?
You want to go ahead and pull it to a stop?
Chris and I are laughing because we were just talking about that.
Copy that.
Total speculation from a marine geologist who likes rocks. And the fact that this little ridge sits close to quite a high relief edge of the shallower flat top of this geo, and possibly the lower flat
coming on top
portion of the geo may have separated from it by faulting. I
think
wonder
we're
if
having a
this
heading change
is
a little more south
an
now.
accident
So
of
we're gonna
erosion,
adjust the
as
ship
some people
into
call them. In other words,
I'm gonna
just
center up and figure out
a
the
chunk
direction.
of the side of the geo that
It looked to me
remained prominent when the rest of it slid down off the edge of the larger feature.
Yeah, I'm going to go to 190.
And if that's the case,
see
then what we may be
like.
looking at may in fact be igneous rocks covered with
It's
manganese
roughly 19,
across 10
195.
stations.
Thank you, Patty.
So, Pilot, you're looking
Yeah,
at...
a few minutes ago I would have definitely said the same thing. It looked like there were some very large blocks.
Yeah, can I use your
I don't know
sonar
what
for a
this
sec?
nice flat stuff is, though,
just
whether
let's do
that's
south.
also
Let's see what that
igneous
gives
and
us.
covered in manganese crust, or if it's something different.
A little hard for them to.
Sirius is not
I
giving
wonder
me a
if
whole
Bruce
lot.
has got the White House contingent in. He would have
So,
pulled it out.
UH, your mic is open?
Hey, Chris, your mic's open.
So maybe 190,
I
roughly.
think they just arrived here, Chris.
So here we can see...
Hi, Bruce. Yeah, thanks. I guess we had our mic open. Yeah,
Right.
we were wondering.
190.
We didn't
Please,
quite
thank
spot
you,
that
Nav.
fish early enough. It was really at the bottom of the screen, but we'll keep our eye out for some more.
I'm sorry, say it again.
You still want 0.2?
Yeah, I think that was another cutthroat eel from Apple-Branca. So there actually have been two of those that have been on the edge of the screen that
think
we've
they can only
traveled
move a
along
decimal
since we
10.
saw the first one. So it looks like that might be a dominant fish species here.
All right, 0.2.
So there's a vase sponge that's in the upper left corner, and that's a different sponge. As we get up there, maybe we can take a look.
Bridge nav. Hey, can we get a ship move of 60 meters, 190 degrees, 0.2 knots, please?
So this is pretty unusual. If we could get some close-ups of this. Not sure I've seen this before.
Pilot, can we get close-ups on this white vase sponge? I figured.
That was 190.
One possibility is it's in a genus Lephroiella, and if it is, we're likely to see anemones all over the surface of this thing. So we'll find out when we zoom in.
Great.
Zooming in. Holding.
So I don't see
I'm
any of
actually
those
gonna rotate starboard
anemones,
just slightly
Chris.
to keep you in view.
So what does that mean?
Just at the heave, you off. I lose you. For a moment.
Then it's another type of vase, and I'm just taking a long look at it before making a fool of myself.
I think just holding here for a little while is pretty good because Chris just needs to get a good look at it, Pilot. Thank you.
Chris, is there a particular part you want to zoom on, like the base?
I
or
guess
something?
we could zoom on the base just to see what it looks like, please, Pilot.
I'm going to zoom out just a bit. I'll hold here.
Is that a squat lobster?
Yes, it is a Kairos-styled squat lobster living commensally on this sponge.
So I do have a guess here. It may not be a very good guess, but I think it's something called, or in the family Tritodictyidae. Haven't seen this very much at all, but the actual general shape of a very elongate funnel like this that flares at the end is characteristic of it. And we did collect one of these in Hawaii. It's a little bit different, but yeah, it's pretty interesting. I'll go ahead and type that in.
Maybe just tilt up slowly. And keep coming. Sure, no, this is fine.
So what you're looking at is you're looking at the small oscular holes in the side of the sponge, and the way this guy feeds is water comes in the side into those holes, and he's got these little beating cells that have little cilia, or not sure that's the right word for it, that are beating and also helping create current through the sponge. And then it goes right through the body wall of the sponge and collects into the center area of the funnel and exits through the top. So that's the game that they play, and they obviously do it very efficiently because sponges have been around at least since the Jurassic.
Do you want to move on? Do you want to move on? Or are we good?
Well, this is Bruce again at IRC. So as expected, the group that was here did a kind of a quick whirlwind tour, told them a little bit about the expedition, the work on the manganese crust environments and deep corals. And they took some of the handouts, but they've gone now.
Okay, thank you, Bruce.
I'm glad we had something for them to look at while they were there.
Now, they had lots of questions about the sponge. They actually wondered what the greenish color was on the sponge, and I couldn't answer that one.
I'm glad you didn't ask me because I wouldn't have known either.
Chris, is this enough imagery on the sponge? Would you like us to look at something else?
Do you want me to keep you in view?
No, this is fine. We can go ahead and move on. I'm sticking with Tritodictyum. That's my guess, and I'm sticking to it.
Okay. Do you want to look at any of these corals?
Zooming out. Full wide.
Yes, let's see. There were some different bamboos. I think I saw 3 of them. I'll tell you what, I'll call one out when I see one that looks different.
Okay, perfect. Well, I'm finding these rocks far more exciting than I thought I would. Much like Patty, though, I kind of wish I knew what I was actually looking at.
Navigator, co-pilot.
So, Deb, don't you think that rocks with decorative branching hats on top of them are more interesting?
We're moving
Absolutely.
190.
I would totally agree. And many of these rocks have a decorative branching hat.
Yes, it looks like 190.
Copy that. Looks like the ship is about halfway through the move.
It is a 60-meter move, and we're a little more than halfway.
Okay.
So for those of you who are just joining along with us at home, or our shore-based scientists who might be signing in,
Copy that.
we are at Pigafetta, Gio. We are at about, we're just a hair over 2,000 meters. We're actually at 1990. We are exploring this ridge feature, which we've been talking about may have actually sort of slid or faulted off of a higher block. And we've already seen a number of different coral species and 2 very interesting sponges, as well as a sea urchin. So it's been pretty interesting just in our first hour or so.
I wonder if we could zoom in on that zigzag sponge. I think it's a Rittigorgia, but it would be good to document it. It's over to the right center right now.
Can you zoom in on that sponge that looks like a zigzag?
Spirally coral, I mean.
Affirmative, yep.
Yep, I think that's where they're going, Chris. Thank you for the zigzag appellation. It seems to work quite well.
Okay, go ahead, video.
I think you might have gone past it.
Is it this here?
Is it that one? I think it must be.
Partial.
Chris?
That looks like a
Yep, this is it.
Okay.
Oh, that's it?
So these are some of my favorite corals, and I think they're favorites also of the pilots and videographers because we got a lot of video of these things. They're simply gorgeous animals. These are kind of small and not quite in their full bloom yet, but Les Watling actually published a paper several years ago where he was describing a specimen of one of these off the northwest Hawaiian Islands as being the tallest deepwater coral that's ever been documented on the planet. So it'll give you an idea of how big these things, they can get up to like 5 meters long, and they're just stunning. They're just beautiful corals.
Chris, did you say 5 meters long?
Yeah, it's huge.
Wow.
It could potentially park right over the ROV.
Wow. And so it looks like we've got some ophiuroids on this coral as well, which we're just taking a look at right now. These ophiuroids are really active.
Okay,
Like they're trying to go somewhere.
Okay, coming wide.
I think we're done with this.
I want to lose the skids.
Yeah, go ahead. Feel free to do that.
The seafloor here has a lot of fissures, which would support the idea that perhaps it is something that was faulted off or slid off of a different area.
I agree with you, Deb. I've been noticing that there are, in fact, some continuous fractures,
Yes.
even all the way across the field of view, and when the ROV tilts up, start moving again, see them going off into the distance. So it is, I completely agree with you. This thing is something that has broken off something else.
There's some there, yeah.
And as usual, for those of you who are interested in the broader context geology, I would highly recommend taking a look at Stream 2, which is the Sirius view, the camera sled view of the D2 ROV. And I realize that I think Diva and I have not actually introduced ourselves on this dive yet. So I'm Deb Glickson. I'm the geology lead here on the ship. I am a hydrothermal event geologist, and I work at Florida Atlantic University Harbor Branch Oceanographic Institute. Diva?
And I'm Diva Amon, and I'm the biology lead on this expedition, and I am currently a postdoctoral researcher at the University of Hawaii.
Thank you. All right, so is there anything we want, shore-based science, is there anything we want to zoom into here? Is there anything particularly interesting that you'd like to see?
That's all you get.
Okay.
Copy.
I already have the feeling this is going to be a great biology dive. I feel like we've already seen a number of really interesting things, and we've only just begun.
You want to come in here, video?
Okay, zooming in to that asteroid.
Yeah. Just while we're waiting here a little bit.
So is that a dead Spock
Hold there.
stalk with a asteroid on top?
Yes, exactly. So this is a dead sponge with a type of sea star on the top of it.
It's kind of a chunky sea star.
Tis.
Chris Ma will be calling in here soon.
Probably will call in. Well, the feed's not very good on shore. So I think this is actually a Echinasterid.
Okay, coming in.
Henricia?
Maybe Henricia, exactly. Henricia pauperima.
Copy. It's got weird.
Yeah,
I think
that's
you
what
nailed it, Diva.
Yeah. I was just going to message you, Chris, to say that I'm relying on you a lot for these corals and sponges that we're going to see.
So are all of its tube feet just sort of pointed up at the end so you can see the little tubes?
No. So no, the tube feet are the things that it's holding on with.
Okay,
which
so
you
what
could
are
just see a tiny bit of sticking off the left. So its arms. Is that what you mean?
Yeah, you know, like at the bottom of each of its arms, it looks like they're tilted up. What are we looking at?
I don't know, I just think it's holding them in a weird way. But
Okay.
so each of its, Deb was just remarking that each of its arms, the ends of them are upturned slightly. But then, because they're upturned, you can just see the little tube feet, which are what hold it in place and help it to walk along the seafloor as well.
Okay, I think we're done here, video.
Okay.
Let's come on.
Coming wide.
Thank you.
So even though the substrate is not that consolidated, there's a lot of boulders and cobbles and things like that, we're still getting a reasonable density of these corals. And if the substrate becomes more consolidated into bedrock a little further up, then I suspect we'll see the density go up even higher than it is now.
So, Chris, these corals really like a hard substrate to be living on? Is that correct?
Yeah, so these larger trees, we saw this in the northwest Hawaiian Islands where these ridges have substantial current running over the tops of them. And if they choose a rock that's a little bit too small, it'll support them initially, but as they grow and get bigger and bigger and bigger, and then the current blows them, it's just like a sail, and it'll blow them right over the flaps, and then they wind up dying on the seafloor. So this type of substrate is all kind of broken up and loose isn't the best for these things. And then if we can get into an area where it's just solid bedrock, then I think we're going to see a lot of these all in one place.
Thank you, Chris. That was great.
Then what was the last one?
From
Yeah,
a
I'd say a
geology
little bit more,
perspective,
maybe,
the ridge is steepest
Yeah, something like
on
that'd be fine, I
the
think.
east side, if I remember correctly. And so if we want to look for more solid substrate, probably the west side of the
Sounds
top
good
of the
to me. I'm
ridge
turning
would
there now.
be a more likely place to go to find that kind of
Can you lose these
impact
skids, your video?
seafloor structure.
Yes.
I'm just flying up a little more today because we got trees under us. I don't want to hit them.
Pilot, that makes a lot of sense.
So I'll be looking through my toes a lot. You can feel free to lose them whenever you want, video.
Copy
Bridge
that.
RV nav. We would like to request a ship maneuver, please. 60 meters at a bearing of 210 at a speed of 0.2 knots.
And pilots, do we feel like we're pretty
Thank you.
like confident that we're sort of in the middle of the ridge, on the top of the ridge?
I will let Nav answer that.
Okay. I will ask Nav then. Nav, this is Watch Lead.
Yeah, go ahead, watch lead.
I was just wondering, do we feel fairly confident that we're like in the middle, on the top of the ridge?
Okay,
Video, let's
can we zoom on that little orange thing that's straight in the center of the screen? I think this could be our first
Bringing
Anthomastus.
us more onto the
Yeah.
top of the ridge.
So, oh no, I think, is it an Anthomastus? I think you're right, Chris.
Oh, okay, sorry. So we seem
Yeah,
to be off
and
to the
Tina's on right
eastern
now,
side
and
of the
she's
ridge
going to
a
be
little
able to write in the
So I'm
chat
bringing us
exactly
more
which
west to bring us on
genus,
top.
at least, and possibly species she thinks this is.
So what is that orange thing?
An Anthomastus
A
coral.
little wide video.
Oh.
Okay, coming wide.
And we're waiting for Tina to give us an even better ID than
Gonna get in
the
close
genus.
here. Yeah, you come full wide. Thanks.
Okay.
Actually, come full wide with the skids, please.
Okay.
Thank you.
It's interesting, again, as a non-biologist, it looks like there are a lot more dead stalks here than we've seen. Is it just because the density of the coral and sponges are higher, and that's why we're also seeing sort of
So
the circle of
co-pilot,
life?
he
I
has
think
to,
you're exactly right, Deb. It's not that
stay off
there's
the
a
bottom quite a bit because
Okay.
of just the number of corals here.
disproportionate amount
Yeah.
of
So if you
stalks.
want to
Just that we're
adjust
seeing so much
swing
more,
arms
so many more
to try to
stalked
get
animals here, so
some
much
more
more
light
corals
down,
and so much more sponges that
that
it's
might
just
be
natural
useful.
that some of them are going to die
like
at some
Okay,
point,
let's
and their
come back to your
stalks
video.
obviously last quite a
Is
long
you
time.
could rotate
Coming
your
back
upper
in.
swing arms, stow them a bit more, so that they're pointing more forward, and you get more light directly down
It's
in front
partial.
of you.
Copy.
Is your back
Yeah, I
on?
see what you're saying.
So that might be one thing to try and see how that helps with the lighting.
Diva and I may have just
The
had a
other one
rousing
is to
chorus
adjust
of the
lower
circle of
swing
life.
arms, deploy
Not
them
on
more,
Keep coming.
screen.
and then rotate them more
coming in.
inboard. But you just want
Without
to be very
hitting-
careful
yeah.
when you
Yeah,
do
just
that.
hold there.
And
Wow,
look,
that's
and have
beautiful.
the utility camera looking at them,
Manipulator.
because you can actually rotate those into the arms. You probably already know that. But
So
you
Chris
can,
is
if you don't
guessing
look at 'em and you start rotating 'em like this,
Anthomastus tehinidus.
you can break the carbon fiber arms, depending on where the manipulators are stowed.
Tis been done.
Yeah, I think Tina will agree that it's definitely
We have stops in there,
Anthomastus,
but
the genus, but
if
as far as the
the ceiling's all
species,
the way
she's
to
probably
floor,
looking
you
at
can
it
see it.
carefully right now, because she knows exactly what to look at
All right.
to really nail it where I don't.
Upper swing arms a little bit.
Yeah, you guys do your thing. Go
Okay.
ahead.
And try to work with video to get an idea of the light pool when he zooms out.
Okay, Watchleys, are we through with this?
And right behind
Yes, I
it,
think
you
we
can
are.
see a small chrysogorgia, and that's the yellow colony in the upper right corner. That's our first chrysogorgia at this site.
We can focus. Crisis.
Oh, yeah.
Where?
Just to the right of the Anthomastus.
Ah,
I'm not going to remaneuver. We'll just try to keep that.
There we go.
boy, the manganese crust on these rocks
so
looks really thick.
Probably throughout this dive.
It does,
we're
and
gonna have to
that's
work with
likely because I
video
think
to
Chris was
assess
saying
the
this
light.
is Cretaceous aged.
Yes, this appears to be a Cretaceous seamount, and
Thank you,
that
video.
means that we're talking
And you can
about
use them
somewhere
again.
between
Okay.
65 million years ago and 145 million years ago. So these are older rocks,
So your upper
possibly even old
your upper light bars
rocks.
are straight or almost more forward, so
Copy. Overhang.
cautious.
Yeah.
Shore-based science, is there anything else you'd like to zoom in on?
Wonder if we could take a look at that that unbranched thing that's in the upper center right now. I don't think we've seen one of those yet.
Can you clarify that?
It's
Watson?
just a small
so in
single
the-
great piece that's
So the whip coral, you see the really tall black
Background.
stalk just in front of that. There's a single white whip coral.
Sort of in the middle center right
Yeah,
now.
Copy.
center, exactly, screen right now.
yeah, and that's correct. And like I said, I'm going to hold off doing close-ups on the big bamboos until Scott's ready to take a look, because I'm really no good at these things.
It looks like there's another one of these up on the top left. Is that correct?
Yes, exactly. Well, it may not be the
Maybe
same
the same one,
species,
but it's another one of these unbranched corals.
Yes.
This looks like a sponge on a coral, but I am no biologist. Come on in, video.
Okay, coming in.
Yeah.
So this
this a
is
coral?
definitely a sponge.
This is partial.
Copy.
Chris, I'm not sure.
It's a sponge. It's a Tritopleura. It's just a younger one where the leaves haven't curled quite as much as the ones we imaged before.
And if we could pan, yes, no,
Pan left and right.
pan
tilt up and
right,
down.
yes. If we could pan right to the dead sponge, pan right
You're on
to
the right.
right to the dead sponge. That's
Is it just behind it?
yeah, it's
very
the black.
tall? Yeah.
yeah, and it looks like it's got a lot of commensals living on it. That just might be quite interesting to see. It looks like this dead sponge has quite a lot more commensals than any of the live animals. So that's just interesting. Do these live animals have, you know, sort of defensive
defense mechanism? Yeah.
to prevent themselves being covered with too many commensals?
I've often wondered that, whether
Come in here,
we've
video.
seen so many with a lot of
Let's roll.
commensals, and I was wondering if it would
Great
affect
snap.
their health
And
in
hold
any
there.
way.
Holding.
Okay, so you can immediately see some comatulid
Great, Jarvie,
crinoids,
now.
as well
Yeah, can we
as,
hold here,
I think
please?
these are
Thank you.
hydroids or bryozoans. Gonna go with hydroids. And then I think I saw some more comatulids further up the stalk as well. Ooh,
What are
We're at
those very...
80
a
degrees
beautiful
south.
shrimp
Copy.
and some barnacles.
Oh wow! We are looking at a clatorizid.
Oh, those are
Another
clatorizin sponge?
This is
Really?
another carnivorous sponge.
Very cool.
Wow.
Stand by, video.
Have you seen them living? Ooh, polychaete. Have you seen them living on a dead sponge before? Dead stalks, Chris?
No, never. I've never seen this one before either.
Does that mean that you're interested?
We need to decide quickly.
We need to make a
Possibly.
decision quickly if you do want to collect it, Chris.
I've already stopped the ship.
Yeah, that doesn't